Plants have cellular receptors that detect changes in their environment. Hormones are chemical signals that coordinate different parts of an organism. Tropisms are often caused by hormones. Any response resulting in curvature of organs toward or away from a stimulus is called a tropism. In the late 1800s, Charles Darwin and his son, Francis, conducted experiments on phototropism and ultimately discovered the presence of plant hormones. In 1913, Peter Boysen-Jensen demonstrated that the signal was a mobile chemical substance. In 1926, Fritz Wendt extracted the chemical messenger for phototropism known as auxin. In general, hormones control plant growth and development by affecting the division, elongation, and differentiation of cells. The first kind of plant hormone are known as auxins, where an auxin refers to any chemical that promotes elongation of coleoptiles. One of the roles of auxin is to stimulate proton pumps in the plasma membrane, known as the acid growth hypothesis. Auxin is involved in root formation and branching, and synthetic auxins can be used as herbicides. Auxin affects secondary growth by inducing cell division in the vascular cambium and influencing differentiation of secondary xylem. The next group of plant hormones are cytokinins, named so because they stimulate cytokinesis. Cytokinins are produced in actively growing tissues such as roots, embryos, and fruits. They work together with auxin to control cell division and differentiation.
Another plant hormone group is the gibberellins. They have a variety of effects such as stem elongation, fruit growth, and seed germination. Gibberellins stimulate growth of leaves and stems. In many plants, both auxin and gibberellins must be present for fruit to set. After water is imbibed, release of gibberellins from the embryo signals seeds to germinate. The next group is brasinosteroids, which induce cell elongation and division in stem segments. Abscisic acid slows growth and induces seed dormancy and drought tolerance. Seed dormancy ensures that the seed will germinate only in optimal conditions. Plants also produce another hormone known as ethylene in response to stresses such as drought, flooding, mechanical pressure, injury, and infection. Ethylene induces the triple response, which allows a growing shoot to avoid obstacles. The triple response consists of a slowing of stem elongation, a thickening of the stem, and horizontal growth. The roles of ethylene include, but are not limited to, senescence, which is programmed death of plant cells or organs, leaf abscission, which is the process that occurs in autumn when a leaf falls, and triggering the ripening process.
Many plants oscillate during the day. Plants that flower when a light period is shorter than a critical length are called short-day plants. Plants that flower when a light period is longer than a certain number of hours are called long-day plants. Flowering in day-neutral plants is controlled by plant maturity, not photoperiod. Photoperiod responses are actually controlled by night length and not day length. Short-day plants are governed by whether the critical night length sets a minimum number of hours of darkness, while long-day plants are governed by whether the critical night length sets a maximum number of hours of darkness. It is thought that plants may detect gravity by the settling of statoliths, which are specialized plastids containing dense starch grains.
Oh no. Where's your email?
Collectively, developmental responses to mechanical stimuli are called thigmomorphogenesis. Mechanical stimuli can also induce a bending response called thigmotropism. For example, tendrils, petioles, and or stems of climbing plants bend towards a touch stimulus and so wrap around supports. In contrast, roots of many species avoid obstructions by bending away from touch stimuli. Environmental stresses have a potentially adverse effect on survival, growth, and reproduction. Stresses can be abiotic, non-living, or biotic, living. Abiotic stresses include drought, flooding, salt stress, heat stress, and cold stress. During drought, plants reduce transpiration by closing stomata, slowing leaf growth, and reducing exposed surface area. The growth of shallow roots is also inhibited, while deeper roots continue to grow. During flooding, enzymatic destruction of root cortex cells creates air tubes that help plants survive oxygen deprivation. Salt can lower the water potential of the soil solution and reduce water uptake. Plants respond to this stress induced by the salt by producing solutes tolerated at high concentrations. This process keeps the water potential of cells more negative than that of the soil solution, thereby negating its effects. Excessive heat can denature a plant's enzymes, so plants have heat shock proteins that help protect other proteins from heat stress. Cold temperatures decrease membrane fluidity, so plants alter their lipid composition of membranes as a response to the cold. Freezing causes ice to form in a plant's cell walls and intracellular spaces.